excited to have you here for this research journal club that is going to focus on what I consider to be one of the most important projects in the bleeding disorders community, at least in the last decade, which is My Life, Our Future. And we've got some really exciting presentations for you. So without further ado, I will turn it over to Dr. Jill Johnsen.
Thanks, Len. I'm going to talk briefly on My Life, Our Future. It's MLOF, and so I think we all know that. I'm going to talk about both briefly what the program did and where it is now as a research resource. And then we're going to hear some really exciting findings that Zuben Sauna and his group have made. Okay, so I'm Jill Johnsen. MLOF was obviously and still is a big program of many people. So I'm one of many people who work to make MLOF a success. And I'm here representing today. Next slide. I have my own personal disclosures are shown here and none are relevant to MLOF. Next slide. So just to get us all oriented, even though we all know a lot about hemophilia, let's make sure we're all talking on the same page and using the same words. So hemophilia A and B are rare X-linked bleeding disorders, which means the genes are on the X chromosome, which is one of the sex chromosomes. Deficiency of factor VIII, which is encoded by the F8 gene, leads to hemophilia A. And deficiency of factor IX, which is encoded by the F9 gene, leads to hemophilia B. The primary complication of hemophilia is bleeding. These are bleeding disorders. In males, this impacts joints and soft tissues and susceptibility to excessive bleeding with trauma and surgeries. We know that females can also be at risk for bleeding, and the pattern may be a little different in them. So that's why we have this caveat that this is the pattern that we best understand in males. Hemophilia severity is defined not by how bad the bleeding is, but by the laboratory measure of the factor activity level, which in males largely correlates with bleeding, but they're not exactly the same. So when we talk about severity, we mean the lab level. So if the activity is less than 1%, then it's severe, 1 to 5% is moderate, and more than 5% but deficient is mild. And another significant complication of hemophilia is something we call inhibitors, which are an immune response that leads to neutralizing antibodies that are impacting the function of the factor. So in hemophilia A, that was previously reported to be, can we go back? Or I'll just say it, 25 to 40% of people would go on to develop an inhibitor. This can also occur in mild disease, and genotype is a known major risk factor for inhibitors, but obviously there are many other inherited and acquired environmental factors that we need to understand. Next slide. So genotyping in hemophilia, there's been a consensus for some time now that the genotype is clinically useful information. It helps reproductive planning, pregnancy and neonatal management, risk of inhibitor formation, predicted disease severity, and helps us better understand what are the mechanisms of disease in hemophilia. And it's a really high yield test to do. 95 to 99% of patients who had been identified to have a variant, a DNA change in the candidate gene if they did genotyping. And also important is that everyone deserves their own genotype. And one of the reasons for that is, especially in severe hemophilia, patients can have what's called a de novo variant, which means that it arose new in the family and there may not be a big family history. So that genetic information can be really important. I'm realizing I'm using the word patients a lot, and that's how we have talked about it historically. And I want to say the caveat here that we do intend to say people living with hemophilia. The genotype is useful in research to understand the causes of disease and complication risk. And if we're ever going to get better, we have to have better high-resolution data to inform the advances of tomorrow. In 2012, only 20% of U.S. patients with hemophilia had a genotype, and that was the unmet need that led to My Life, Our Future. Next slide. So what was My Life, Our Future? It was a U.S. national project to genotype patients with hemophilia and to establish the research repository we have today. MLOF formed as a partnership of Bloodworks Northwest, where the PI, Barbara Conkle, was. The Athens, the National Hemophilia Foundation, and funding came from Biogen Bioverativ. And additionally, MLF was performed, and most importantly, probably in a lot of ways, in collaboration with a huge number of hemophilia treatment centers that made this work possible in a rare disorder. And obviously, with those staff in, also absolutely essential is the participation of patients, at-risk females and family members. MLOF developed a research repository. Participants were offered the ability to consent to research, and a high percentage did. And from them, we have clinical data linked in Athens and what was submitted to the at the time of genotyping, and biological samples. As part of that consent, they consented to whole genome sequencing and dbGaP, which is an NIH shareable resource, and that made access to federal funding also possible, that has enriched the research repository. And really, the big goal is to support the research community to do research to give back from this project to people affected by hemophilia. Next slide. So over 11,000 people participated in MLOF. We looked at the demographics of MLOF participants relative to U.S. HTCs, which were the sites that enrolled, and they were all very similar. So you can see it was about 4/5 hemophilia A, 1/5 hemophilia B, which is approximately the relative proportion in the populations. We sought to enrich for female participation. So you can see there were some more females than males than the HTC population. The hemophilia B population was numbers were vastly helped by significant outreach in the later years of the project to get the cohort representative. And you can see there's a lot of pediatric patients, small kids, and that this also was largely representative of the HTC population. The demographics for race and ethnicity were also very similar. Next slide. So we believe we have a large and representative cohort. We looked at the DNA variant type that we found in the genotype, which is basically that kind of DNA change that was happening. And here we see the proportions of the different kinds of genotype that we saw, the different kinds of DNA changes by which kind of hemophilia and how severe, how low the levels were. So on the left is less than 1% severe, in the middle is moderate, 1 to 5%, mild is on the right, greater than 5%. The top row is hemophilia A, and the bottom row is hemophilia B. So one of the big take-homes is missense, which is where a DNA change causes a change in the amino acid in the protein, is in orange, and that's the predominant type in moderate and mild disease, and still a big contributor to severe disease. But additionally, in severe disease, we see in the gray wedges inversions in hemophilia A, and we see other big impactful kinds of changes. So yellow is what we call large structural variants. Those are big shufflings of the DNA. And we see other things like something we call gain of stop, which is where the variant has told the protein to stop making itself, and those are in red. And those are predicted to be severe, and they are in the severe category, which really helped to validate that what we were finding in MLOF makes sense in hemophilia and validated many other prior earlier studies. Next slide. So we're here to talk about inhibitors today. So here's what we saw. Thanks to the American Thrombosis and Hemostasis Network data set, there was inhibitor data on a significant number of participants. And here's the data from hemophilia A relevant to the Journal Club today. So next slide, or next click rather. There you go. So you can see that we had about a 30% inhibitor rate in severe A, similar to prior smaller studies, and about a 9% inhibitor rate in mild to moderate hemophilia B. I want to note that the inhibitor data was really binary. It was yes, no, which really doesn't capture the full complexity of inhibitors. So that is one limitation here. And another limitation is that we were relying on the sites to give us an accurate severity. And we have some individuals where we're pretty sure they had severe, but the site listed them as 1% rather than less than 1%. And when that happened, we put an asterisk on the right. So next click. So you can see we have this category of pretty high risk genotypes. The first one is something we call SVDEL. That just means there's a big deletion of sequence. The sequence is just missing. It makes sense that they probably don't make a protein, and they're very likely to have an inhibitor. And then we have these two new categories of high risk inhibitor. One is something we're calling a complex inversion, which means that because of the way we sequenced an MLOF, which was relatively new, we not only looked at inversions, but the rest of the gene. And we found out that there's a group of people that not only have an inversion, but have another big gene change. And those people were at high risk for inhibitor. And then another new finding, and a little bit unexpected, was what we call NVF, or no variant found, which means we threw everything the clinical lab had at these people and a little bit of research, and we could not solve them, but they had less than 1% factor activity level. And that was also associated with a high inhibitor rate. And I can say in preliminary news, we're looking to solve these with additional research. And so far, it looks like it's because they have a big change elsewhere outside of the area that we genotype. Next slide. Then we have the inversions, the gain of stop, and they are all about 1/3, 33% or so, which is sort of consistent with prior studies. Next click. And then we have missense variants can still be associated with inhibitor development. And you can see both in severe, but also they're the predominant type to develop inhibitors in non-severe hemophilia. And that's probably because they're changing something that's recognized by the immune system that's different between what the person makes and what their factor replacement product looks like. Next slide. In hemophilia B, similarly, we saw about 12% inhibitors in severe hemophilia B, again, consistent with prior reports. And also, again, big structural variant deletion is associated with about over 50% inhibitor rate, similar in hemophilia A. And then we did see some with mild to moderate hemophilia B, and again, those were missense, but those were pretty unusual. Next slide. Oh, this was just highlighting what I just said. Next slide. And there we go. Next slide. We also were able to look at hemophilia inhibitor rate, race, and ethnicity. This is using self-reported race and ethnicity. And there's some another recent paper in JTH I'd point you to by Kate Pratt's group. So this is an aggregate across the entire spectrum of everyone who participated, who had inhibitor data. And in it, we found that relative to people who self-reported as white, anyone who reported as Black or African-American in that category, both in hemophilia A and in hemophilia B, they were at statistically significantly higher risk to develop inhibitors. In the way that the analysis was done, we don't see statistical significance for other categories, but I want to point out that quantitatively, just based on the number, Asian Americans may also have higher inhibitor rates, and I think more work is needed. Unfortunately, there's no more resolution on what it meant to be of Asian ancestry, which we know is a really diverse group. In addition, we were able to look at ethnicity, people who designated as Hispanic, Latinx, or Spanish origin versus whether they have that or not. And that's independent of race. We found a statistically significant signal that being of Hispanic origin was associated with higher rates of inhibitors, both in hemophilia A and hemophilia B. And I'd like to take a moment to point out that race and ethnicity are a really complicated thing to both pick where you are in the category, and also are undoubtedly a mix of genetic and socioeconomic determinants of health. So I think this tells us there's lots more to do to better understand what is driving the signals here in inhibitor development. Next slide. So, I apologize, the animation, so it's hard to do alone. So this is the difference between people who self-reported as white and Black. And then the next slide will be highlighting the difference between those who reported as Hispanic or non-Hispanic origin. Next slide. So in summary, MLOF provided clinical genotyping to over 11,000 participants. This makes it by far the largest hemophilia genetics project ever performed. We estimate about 25% of the U.S. male hemophilia population was participating, and we captured about 40% of those that are followed at HTCs. We found a clinically reportable DNA change in over 98% of male patients who had hemophilia. This is a very high yield genetic test, and our method is a very comprehensive method and supported the need to use a comprehensive sequencing method, because we did find people who had more than one DNA change, which is really impactful, particularly for females. The discovery rate was really high to find something that had never been seen before. So we classified a novel variant as not being known in hemophilia, never reported, never found in a clinical lab, and not a common variant in the population. And over a third of variants that we found in MLOF were new, and we kept finding them up until the end of the project. So that really says we're not done to understand what are the genetic changes that cause hemophilia. We also resolved some questions about genotypes related to high-risk inhibitors, but also opened up many other questions. We identified these two new high-risk genotype categories, which are the complex inversions, and when we don't find anything at all is an important category in severe disease. And we also report that inhibitor risk differed between self-reported race and ethnicity groups. Next slide. So what is the MLOF research repository? One of the big goals from the very beginning of MLOF was to return findings to the community and build a resource for the scientific breakthroughs of tomorrow. So as far as returning findings to the community, all MLOF genotypes have been deposited both in the CDC and EHAD databases and are publicly accessible. There's no way that you can figure out who someone is. It's very protective of privacy, but the most important pieces, which are the gene change and what kind of hemophilia was associated, are there. In addition, we have done ACMG variant classifications, which is interpreting the significance, which will be made available publicly. And I'll just say from memory that we also have developed this research repository, which is not only got data and samples and the potential to be linked to data in Athens, it also has genomic and other omics data being developed in partnership with NHLBI, who have accepted this cohort into their TopMed program. Over 9,000 samples have whole genome sequencing, and over 4,500 will have whole blood RNA sequencing. And it's supporting research through applications. We have had 2 cycles of researchers approved, and one of them is Dr. Zuben Sauna. So I'm really looking forward to him sharing his paper. And many acknowledgements for MLOF, this slide and many others. And thank you to the entire community.
Can I start?
And now we will hear from Dr. Zuben Sauna. Thank you. Thank you again, Dr. Johnsen.
Thank you for inviting me. And as a user of this data set, I am immensely grateful to the My Life, Our Future project. My name is Zuben Sauna. I work in the Food and Drug Administration. I have a regulatory role in that. I am the director of the Division of Hemostasis, but I would like to emphasize that we also have academic research interests, and my presentation today has nothing to do with my regulatory role. Could I have the next slide, please? Before I start, I would like to, at the onset, express my deep gratitude to the individuals in my lab. I'm going to be presenting the work that has primarily been conducted by Vijaya and Joe, but I am equally grateful to the other folks who have done a lot of other, very, very interesting work. And if you could go to the next slide. So, we regulate a wide range of replacement proteins, monoclonal antibodies, etc. And I would like to put the factor VIII molecules in the context of therapeutic proteins as a whole. And if you see today, of the top 10 best-selling drugs, 7 are biologics, and in terms of U.S. biologic sales, the numbers have shot up considerably in the last decade or so. However, that's not solely what's important about biologics. These biologics invariably address unmet medical needs that other products cannot. They provide cures or permit the management of extremely complex diseases. And as this community well knows, they improve the quality of life considerably for millions of people. Could we go to the next slide? That being said, biologics have one significant drawback which small molecules do not. They can be perceived as foreign and elicit an immune response. And when anti-drug antibodies develop, these may or may not affect the activity of a drug. Antibodies that affect drug activity by binding to active protein domains are called neutralizing anti-drug antibodies. In this community, we also call them inhibitors. Non-neutralizing antibodies are not necessarily benign either, as they can affect the PK/PD, the activity profile, and cause loss of tissue targeting. And in the worst case scenario, anti-drug antibodies can also cross-react with endogenous proteins, which in some instances may be partially functional, or they could elicit anaphylactic reactions. Can we go to the next slide, please? So immunogenicity is a problem not only during drug development, but also in the clinic. Regulatory authorities pay considerable attention to potential immunogenicity of protein therapeutics. But like I mentioned earlier, most of these therapeutic proteins address serious unmet medical needs. And at the end of the day, taking the totality of the evidence, the overarching risk-benefit evaluation skews in favor of drug approval. And on this graph, you see several different classes of drugs, and you can see that the number of patients developing anti-drug antibodies in the clinic is quite considerable. And now we are coming to a new age of novel modalities, gene therapy, CAR T cells, gene editing, etc. And that's largely an unknown with respect to the level of immunogenicity, but we suspect that it will play a significant role in many of these modalities because we often use products which are derived from either human pathogens or microbes or so on. Moving back to hemophilia, as Jill's presentation showed you, approximately 20% of hemophilia A patients and 30% of severe hemophilia A patients developing neutralizing anti-drug antibodies or inhibitors at some point in their lives. So this is an issue that unfortunately could not be resolved during drug development, and we have to deal with it in the clinic. Let me go to the next slide. So this is just in the paper that I am going to be talking to you about, which is HLA variants and inhibitor development in hemophilia A, a retrospective case control study is using the Athens dataset. Go to the next slide. So let me just briefly touch on the major players in immunogenicity and the level of complexity that they bring to the problem. So first of all, you have the antigen, that is, in our case, the drug that we are talking about. And depending on the size of the antigen, hundreds or even thousands of peptides can potentially be generated and presented to the immune system. Then you have the protein processing machinery, in that the antigen or protein has to be processed into peptide fragments, and not every possible peptide can actually be generated in real life. And then these are presented to the immune system by the major histocompatibility complex. And this is the big elephant in the room. The MHC is polygenic, that is, every individual contains several MHC genes. The MHC is also polymorphic, and the population has variants of each gene. And the MHC genes are the most polymorphic in the human genome. And to compound it still further, each MHC molecule binds different peptides, often derived from the same protein or the same area of the protein, with very different affinities. So this is the scenario that we have to deal with. Could we go to the next slide? So this is just to give you a flavor of the role that HLA could play in developing an immune response. And this is a very old paper from a very small data set, but the point that I'm trying to illustrate is the two major genetic factors that are at play when you develop an inhibitor to factor VIII. And what I'm showing here are AUC curves, and it's talking about these two variables. And if you have an AUC of 1, that's an almost perfect prediction, if you will, and an AUC of 0.5 is no better than random. And if you just look at the factor VIII sequence, and this is for a few patients who have been genotyped, and you look at how different the endogenous factor VIII is compared to the drug that is being infused into them, and use that as a predictor in terms of some measure of what is the level of mismatch, you have an AUC of about 0.72, which is quite good. Now, if you HLA type each individual patient, you take the foreign peptides that each individual patient is seeing as foreign and look at their affinity for the HLA of that patient and use that as a predictor, you go to an AUC of almost 0.9, which is a very, very good predictor. But, caveat, this is a very, very small data set. So could we go to the next slide, please? So in terms of the study I'm going to be talking to you about today, this is a retrospective case control study, and data from the Ethin data set was merged with the HLA typing data, which we obtained for 997. We aimed to do it for 1,000 participants. A few participants, we couldn't get the HLA typing. And statistical analysis was performed as a series of univariate logistic regression models that would determine inclusion of a variable in a multivariate logistic regression model. Next slide, please. So first off, there have been several studies, often with smaller number of individuals who were HLA typed. And before we venture into how many individuals do we want to HLA type from the ones who have been genotyped by the My Life, Our Future, we tried to get a rationale for the number that we wanted. So based on the probability distribution of HLA variants, we created distributions of HLA variants in patient samples. These are virtual patient samples ranging from 100 to 1,500 patients. And the model was run 100 times for each sample size. And the requirement for this was that each HLA occur greater than 30 times for statistical analysis to be valid. And we found that for a sample size of about approximately 1,000 patients, adequate coverage of HLA variants can be expected in all 100 runs. So you see these, if you can see my cursor, over here you see that these are, you have very little variation. Almost all the patients capture what we need. And this green bar shows us where we need to be. And these orangish, red, whatever color you want to call it, bar shows where we are with most clinical studies in hemophilia A and where we were with studies that were based on HLA typing. So we were way off where we needed to be in terms of sample size. And this is about the bare minimum that we are talking about for any meaningful statistics to be done. Can we move to the next slide, please? So just very quickly, as Jill just mentioned, we have a very many more number of samples, but at the point that we started using the My Life, Our Future data set, there were, we received about 7,151 samples, and then there were, various reasons why we, cut down on the samples, and ultimately we obtained 1,000 DNA samples were requested, and prioritizing inhibitor positive patients. Could we go to the next slide? So one of the first things that we did in our HLA type group, we compared it to the entire Athens data set. And we looked at things like disease severity, primary treatment type, race, ethnicity, age, variant, the mutation that was there in these. And you can, this is published material, so you can pull it up. And so the essential conclusion was that, of course, there was some variation, but there was no major bias in our cohort compared to the our cohort of 1,000 compared to the full data set of over 7,000 individuals. Could we go to the next slide? Now, then I brought up the issue with the diversity of HLA variants in the human population and why that is so important. And therefore, we wanted to check that in terms of not only variant diversity, but the frequency at which these different variants occur, they are more or less comparable to the North American population. And we found that the HLA-DRB1 alleles identified in the 997 participants cover 99.5% of the allelic variation in North America, and 18 alleles representing 82% of the North American population were actually identified at least 30 times, which was our aim, which was our overarching goal to start with. And could we go to the next slide? So in terms of the methodology, all variables were first analyzing using a univariate logistic regression model, and those determined to be significant were included in the final multivariate model. Based on variables selected using univariate analysis, a final multivariate model was fit. The variables for the multivariate model were 18 HLA alleles, 1 variable for race, 4 factor 8 variant types, 3 variables for age, and p-values for variables were adjusted, and results with a false discovery rate of less than 0.05 were considered significant. Could we go to the next slide? And this is an overview and a table form of the multivariate analysis for all HLA typed participants. And we will see them in a more easier to read format later. So if you could move on to the next slide. And this is, again, the same kind of analysis done with a subset of only severe HLA typed participants. And could we go to the next slide? And and this is, in a figure. So this is, whatever popped up as, significant, and along with the odds ratio that, so among severe HLA type participants, we found ethnicity, that is, if you were Hispanic, you had a higher odds ratio of developing anti-drug antibodies. As Jill mentioned, even at just, cursorily looking at the data sets, that large structural changes where an individual hemophilia patient is losing a large amount of their endogenous factor VIII, that is a high risk factor for developing anti-drug antibodies. Missense variants, actually, you are less likely to develop anti-drug antibodies because you have, just one amino acid change. You're essentially making the rest of the protein. And you see one HLA DRB10202, which has a significantly higher risk of developing anti-drug antibodies, and a couple of alleles where you have a lower risk. Actually, they are protective in terms of developing inhibitors to factor VIII. And if you look at all HLA type participants rather than just the severe, you find more or less the same pattern, except that we no longer see the protective HLA alleles as being that protective in this analysis. Could we go to the next slide? So our conclusions are that among participants with severe hemophilia A, we determined that only one HLA variant was associated with a high risk of inhibitor development with an odds ratio of 16.5. Two variants, DRB1 0407 and 1104, were associated with a lower risk of inhibitor development, and similar trends were found in data examining all HLA type participants, and the two seemingly protective HLA-DRB1 alleles failed to reach any statistical significance. Could we go to the next slide? Now, I would like to reiterate the importance of a representative set of HLA variants. So HLA variants identified as having a significant association with inhibitor development actually were found to be relatively rare in the North American population. The HLA-DRB1 0202 occurred in only 10 participants, which is less frequently than most other alleles. HLA typing of a relatively large cohort of participants allowed a rare HLA allele to be identified in sufficient, at least minimal numbers, to do any kind of statistical analysis. And could we go to the next slide? Comparing our results to previous studies, we failed to confirm findings in previous studies on associations between a considerably larger number of HLA types and inhibitor development. And the plausible reasons for that are small sample sizes in previous studies, reliance on univariate analysis failing to control for correlation with other cofactors. Previous studies may not have included a representative distribution of HLA alleles, and given their sample sizes, it is very likely that they did not. And finally, smaller studies unlikely to include all HLA variants or sufficient replicates of each variant for any statistical analysis. And could we go to the next slide? So, so this is, I have given you, presented the results using a statistical approach to try and, tease out meaning in the sense, or in the sense of we were looking at, the role of HLA. So this whole study was primarily, geared towards looking at the role of an individual patient's HLA as a standalone variable. And, I'd like to distinguish between statistical approaches and newer machine learning or AI-based approaches, where in a statistical approach, we try and deduce relationships from data. Whereas in a machine learning approach, it's more hypothesis free, where we try to learn patterns from the data itself. Again, in the statistical approach, we fit the data through a probability model, and using machine learning, we can make robust, accurate, data-driven predictions. And in the statistical approach, we infer relations between variables from data. And in our machine learning approach, we find actually new insights and patterns in the data itself. And finally, the statistical approaches seek to investigate correlations between different variables which we decide on. And the machine learning approach seeks to make predictions from data using various learning algorithms. And we've just completed a study using a machine learning approach, again, using the My Life, Our Future data set. And it's in, I think as of today, it's actually published, and here is the reference. And we go to the next slide. So we've been struggling with the problem of inhibitor development and inhibitor in hemophilia and in other proteins as well, to be fair, for decades now. And I'd just like to end with this little bit of pop philosophy from this the Dev Patel character in this movie, who says that everything will be all right in the end, and if it's not all right, then it's not quite yet the end. So with that, thank you for your attention. My email information is here, and anybody is very welcome to contact me at any time. And thank you again for this opportunity to present this, very, very involved and very educated community. Thank you very much.
Thank you for that presentation, Dr. Sauna. We now have Len Valentino, the President and CEO of the National Hemophilia Foundation.
Yeah, thank you. Zuben, thank you very much for the great presentation. And Jill, same to you. Thank you for the presentation. We can open it up for a few questions. We have a few minutes to be able to do that. If anybody in the audience has questions, we can definitely do that at the end, but we'd like to turn now to our lived experience experts. So we have two people here, Sarah Ayers and Mosi Williams, to talk about their experience with My Life, Our Future and what it meant to them as people living with a bleeding disorder. So Sarah, would you like to go first?
Hello, my name is Sarah. And for us, we wanted to join My Life, Our Future because my grandfather died when my mom was 6 months old. So we didn't have the understanding about hemophilia. And if we could help out future, people, that is what we really wanted to do. And it's nice to actually see kind of what all the research has come through and to see that they have been working on it and finding new, I guess, just kind of how everything works. That was our main goal. So we can't wait to see what the future brings.
So Mosi, would you like to provide any comments?
Sure, thanks, Len. Hi, my name is Mosi Williams. I'm a patient with severe hemophilia A with an inhibitor. The reason that I decided to participate, similar to Sarah in a way, is like wanting to, looking towards the future to help others. But when I was first diagnosed with an inhibitor, I think I was around 10 or 11, and it was a scary experience because I just, this was in the the late 80s. And suddenly my factor wasn't working. And they said, you have an inhibitor. And I didn't know what it was. I didn't know anyone else who had one. And so not understanding that, and then eventually going through immune tolerance induction to where I found a level of factor that I could take, factor VIII treatment that my body would accept. And going through that process and wondering, what the future would look like. And for other patients with inhibitors, particularly with NHF, I participated in the inhibitor summit. So we have all these families and we're all learning together about what it means to have an inhibitor and just the scary parts of some of the dealing with that particular aspect of hemophilia. And so I just wanted to learn more about it, what possibly could cause inhibitors, and then what they could do in the future to, if possible, eradicate it. And so it's encouraging to see this research and also even possibly involving AI that can look at the numbers in ways that researchers may not currently see. And then with some of the things that are on the forefront in terms of gene therapy and other treatments for inhibitors, it's an exciting time.
Thanks, Mosi.
Thanks so much.
So I'll turn it over to Mike Recht, who's going to handle our Q&A period here.
Great, Len. Before I go to questions, I do have one more question for Mosi and Sarah. And when you see these presentations by Jill and Zuben, can you reflect a little bit on the impact it has on you? Is it understandable? Do you see where this research could lead, how it's impacting care right now? Because these are very complicated scientifically.
Yeah, I know for me, it's a little hard to understand, but you guys do explain it quite well, breaking it down for us. I know for even like with my son, he has 2 mutations for hemophilia A. So that kind of confuses me even more going,
Right,
I only hear people talk about 1.
right.
My son has 2 and his hematologist, everybody here, they just don't seem to care. And I'm over here going, he started at 10% at 2 and now he's at 4. He keeps dropping and he's now 14. So wouldn't that kind of have something to do with it? But a lot of people, it just seems like it doesn't matter. Once you get a diagnosis, it doesn't matter. For us, it's more, I want to make sure he's okay. We don't have the family history like most people do. We have, everybody passed away in 1961 and in the 50s. Like
Yeah,
they didn't live long. Now we're seeing how much research came from the 60s to now is remarkable. So I can't wait to see what happens in another 40 years. So for us, we love doing the research stuff. We could do, like, we would just be like, here you go, take all the blood, figure out what you need, because it helps us going, there's going to be a better future for these kiddos later on, even if it's not my child's, maybe possibly his kids or his grandkids. There could be something you guys could find that would work for each, patient, not just go, you have hemophilia, this is your treatment. Maybe it could get specialized, saying this one didn't work, try this, live a
yeah.
good, healthy life. That's our hope.
So just to reflect on what you're saying, I think what I'm hearing is the potential for using this kind of information for a more personalized approach to therapy.
Yes, personalized would be amazing because we're used to not getting any treatment. My son was, 4% and had 5 joint bleeds, and I had to beg for factor just to get on prophy. They just kept saying he's fine.
Yeah,
It's like my son had to suffer so long. So I hope for future patients, they don't have to suffer. They can just be looked at, I have hemophilia, let's go, not
right.
worry about the severity. Just look at each patient as a person, not just numbers. Look at
Yeah,
how they bleed.
I really appreciate that approach. And that's something that we at the NHF, as part of the National Research Blueprint, are really taking seriously. This idea of mild, moderate, and severe hemophilia based on a number versus mild, moderate, and severe hemophilia based on the human being that's sitting in front of you and their bleeding pattern. Yeah. Mostly when you reflect on the 2 papers that we learned about today, do you see how there could be a connection to therapy that might improve for you?
Yes, I mean, it was a bit above my head, some of the discussion, understandably, but I mean, I could see, and the consideration of is in terms of diversity, equity, and inclusion, looking at minority groups, and particularly as an African American male, that there seems to be somewhat overrepresentation in inhibitor numbers. And so the fact that, looking at that and seeing those differences and wondering what might be possibly involved in that, genetically or otherwise. So that was encouraging to see that those things are being pursued and considered.
Great. Thank you. Thank you very much for that input. From the audience, I'm going to pose this to both Jill and Zuben. Is there any research on gene deletion in the person being tolerized for inhibitors? So I don't know exactly what this question is answering, but maybe does the gene variant predict success with immune tolerance?
Yeah,
Do we have any data
this
about
is
that
one of the important questions. So there is one small study that I'm aware of that grouped the genetics into 2 categories, which were high and low risk. So I think there's some caveats in that paper, and that we really still kind of don't have it completely nailed down which ones are high and low risk.
yet?
But we kind of have a general idea. And then they looked, these were all patients with severe hemophilia A who had high titer inhibitors and then did low dose ITI. And the success rates were really different between the genotype classes. So these were people that already went ahead and got an inhibitor and then went on to their treatment. And it really suggests that even when there's what looks like the same thing with regards to titer, that they're probably not the same thing. And that the cause of the hemophilia underneath is probably leading to a different immune response. And so this is the one piece of evidence I know of that suggests that that's the case. There's other evidence that suggests that the kind of inhibitor you make and how many different parts of the factor VIII molecule your immune system sees also is related to the genotype. And the deletions kind of seem to be the worst of all of them. And it's probably because they have no way to make a protein without any part of the recipe, or they're missing a big chunk of the recipe for the protein. So that probably underlies, in that big table I showed where there's a difference by the kind of variant that made the factor VIII gene not work. There's lots of different ways to make factor VIII not work. Within each of those categories, there's undoubtedly more granularity that we need to understand to get to what we really want, which I totally agree with both of our leads today, is we want to get to the point where an individual person in front of us can be informed by this data, and whereas right now we have group level data. Yeah. Deletions are probably one of the categories where it's safe to put big deletions all together, but in some of those other categories, it's going to have a real nuance to what it's doing to the protein. So that's what I know. There's not much. We need to keep doing work.
Zuben, do you think that you could apply a similar framework using machine learning to help predict success to ITI for those who do develop inhibitors, knowing the genotype?
It depends on what data elements we get, and the quality of, because often we by the time we culled, and the numbers look good to just before I'd like to state that both these, like if you use machine learning or AI or any of the other models, numbers are important. I mean, where it's really, really successful, compare, just for a moment, think about the number of data points that Netflix or Amazon has when it's trying to predict what you want to buy next or watch next, compared to what we have in this case. And again, like when you have immense numbers, it doesn't really matter if the quality, and I don't mean in quality that the individual researchers haven't done a good job collecting the information, but having all the information for all the individuals in the data set is very critical. But I would just like to segue a little bit from that in terms of, in terms of immune tolerance, in addition to predicting or looking at who might benefit from what. I think there's a lot of now a lot is exaggeration, but there's considerable data coming out that seems to suggest that the FC fusion protein is better at inducing immune tolerance than the factor VIII molecules themselves. And now we have just a few months ago we approved another variant from Sanofi, what was Bioverativ, which is a even more complex molecule, which has a von Willebrand factor along with the factor VIII, and that is also an FC fusion protein. And there is a literature emerging which not only tells you that in the clinic these do better, but there may be some mechanisms by which these kind of molecules are actually able to selectively, target the specific B cells that are making inhibitors to your particular protein of interest. And there is actually, in addition to this, there is a lot of research coming from the fields of autoimmune diseases, where people want to go beyond just just like we're I completely agree with your notion of therapies being targeted to individuals, but in the same way, in terms of immunology, we are so broad-based in trying to completely suppress an immune response compared to targeting a recipe to kill the immune response that is detrimental to you. I mean, so I think there is a need for looking at this more broadly. On one hand, looking at the individuals and predicting the risk factors that make someone more prone to develop inhibitors, but also bringing in all this new information from diverse fields where we go beyond just hitting somebody with large quantities of the factor VIII and hoping for the best kind of strategies. So
Great, thank you. Well, unfortunately, because it's 3 o'clock Eastern, we do have to call this session to a close. I want to thank all the panelists who joined us today. Mosi and Sarah, thank you very much for your insights. Dr. Johnsen, Dr. Sauna, amazing work. I really appreciate everything that's gone into what I agree with Len is one of the most important projects that has been in our inherited bleeding disorders field in many, many years, and looking very much forward to finding out new information as the data matures and people start thinking about how exciting ways to use it, like you're thinking about, Dr. Sauna, with the machine learning and AI. So thank you very much, everybody, for joining. And Dr. Valentino, thanks for being there and including your thoughts.
thank you.
everybody. Thanks.
Thank you.